Hello, dear students. How are you? I'm audible clearly, Jovita and Jaslyn.
Sir. Yes, sir.
So today, in the previous class, we were discussing the monohybrid cross. Okay, and what were the conclusions of monohybrid cross that we have discussed? Tell me what what were the conclusions of monohybrid cross? Tell me.
Pollination.
I'm asking that what were the conclusions of monohybrid cross? What were the conclusions of monohybrid cross? The monohybrid cross tells us that for it gives in monohybrid cross we have two conclusions. One is the principle of dominance we have. Okay, and the second we have law of segregation, law of purity of gene. What are these two laws states? These two laws state that okay, that whenever in heterozygous condition is there, capital T and small t was there, the one gene is dominant over the other gene. Okay, are you getting my point? That one gene is dominant over the other gene. Okay, understood what I'm saying?
Yes, sir.
That one gene is dominant over the other gene. When there is a heterozygous condition, that in that condition, one gene is dominant over the other genes. And what is law of purity of gene says? That in the F2 generations, okay, we are having for every 4 plants, we were having 3 talls and one dwarf plant was there. So here in heterozygous condition, the small t gene is also there. But they are not intermixing. We are not getting a 3.5 feet plants, or we are not getting in an average plant. Like we were having 6 feet plant in F1 generation crossbreed with 1 feet plant. So we were getting all 6 feet plant that were having a gene of capital T and a small t, capital T and a small t, capital T and a small t, and capital T and a small t. Okay, they are one tall. So this shows that one gene is dominant over the another. In heterozygous condition, one gene is dominant over the another. This is the conclusion that we get in the monohybrid cross. A second conclusion we get that the genes are not intermixed. They maintain their identity. Here, when small t is there, it gives you dwarf plant. It gives you dwarf plant. Okay. So genes are not intermixed. Okay. You understood? Genes are not, tell me, they are not intermixed. Like they are not going to give you a small and average plants. When a gene of capital T and small t, they are there, they are not getting you the intermixed gene. Okay. They maintain their identity. They are not intermixed. They live together, but they are not intermixed. Okay. This was the conclusion of monohybrid cross that we have discussed in the previous class. Okay. Got it?
Yes,
Got it?
yes,
Got
sir.
it? Next is in the dihybrid cross. When Mendel was doing the dihybrid cross, then during the dihybrid cross, actually we were having a round yellow. Okay, in the dihybrid cross, when Mendel was doing a dihybrid cross. So what is actually dihybrid cross? First, let me explain you that in a dihybrid cross, the two contrasting characters or two pair of trait is studied. Two contrasting character or two pair of trait is studied. Okay, what is two contrasting character? If I talk about two contrasting character means that if it is round, okay, if it is round, one is shape and another one is the color. So round, color and wrinkle, round shape and yellow color. Okay, if I talk about round shape and yellow color, both are studied simultaneously. Got it? Got it? You understood here? Understood that what is dihybrid cross? Actually, dihybrid cross is the cross in which the two traits, the two pair of traits, traits or two contrasting character. Two characters are studied simultaneously. Two pair of traits means for every character we have at least two traits. For example, shape is one character and color is another character. The two characters we are studying simultaneously. And in this shape, we have round shape and wrinkled shape and wrinkled shape. And in color, we have yellow color and green color. Green color. So what is mono dihybrid cross? The cross in which two characters, one shape and one color, or two pair of traits. Two pair of trait. One pair of trait is this round and wrinkled. Another pair of trait is yellow and green. So the trait in which we studied two characters, two contrasting characters, or two pair of traits. So please note it down. Note it down the definition of this one, please, everybody. Note the definition. Done. Dihybrid cross. Note it, please. So the characters, the cross in which two contrasting characters or two pair of traits are simultaneously studied. Studied simultaneously. Add one more word. Simultaneously. Simultaneously. What is happening here? Okay, noted. Done. Next
Yes, sir.
is, okay. So what we are going to have here is that he have in F1 generation, for example, first he take round yellow, one seed with round shape and yellow color. Okay, round shape and yellow color. And another with wrinkled, wrinkled shape and green color. When he cross pollinate this one, keep in mind always that in cross pollination, we obtain the same results. Like in the monohybrid cross also, when we were doing capital T and small t, we were having all same traits. Okay, are you getting? We were having all same traits. Okay, same tall plants we were obtaining. This is capital T, small t, capital T, small t, capital T, small t, all were tall plants. So in F1 generation, we were having all tall plants. The same case is for here also, that in the monohybrid cross or in the dihybrid cross, when we were doing the cross pollination, we were obtaining the same results. Okay, this will help you to obtain that which type of pollination is being done here. So it is a cross pollination or the self pollination. So if we are getting the same results, same type of plants, okay, means if we take round yellow and wrinkled green, so wrinkled green is absent. There is no wrinkled green here. All are round yellow. So it means all are same. So if all are same means cross pollination is done. Keep in mind. So note it down. When he when he crossbreed with round yellow and wrinkle green, then he get all the round yellow. He get all the round yellow in F1 generation. But it was capital R, small r, capital Y, small y. And here is capital R, small capital Y, capital Y. So note it down. When same result is obtained, then okay, then it is obtained by cross pollination or in F1 generation. In F1 generation, note it down. This is F1 generation, or we called it first filial generation. Note it down. First filial generation. Note it down, please. Noted? Noted? Madiha, Jaslin, Jobita?
Yes, sir.
Yes, sir.
Okay. Now, what happens in the dihabit cross? If I talk about dihabit cross, when this one is self-pollinated. Sorry, yeah, this one is self-pollinated. Means this generation is going to be self-pollinated. Then what is going to happen? Okay. So let me explain you that what is going to happen here. This is F1 generation, which is round yellow. When it is going to self-pollinate, then we will have 9 round yellow in F2 generation. This is the result of F2 generation. So in F2 generation, we're going to have 9 round yellow, 1 wrinkled green, okay, and 3 round green were obtained and 3 wrinkled green. You check it. We only take round yellow, we only take round yellow, we only take wrinkled green, but we didn't take round green and wrinkled yellow. We don't take wrinkled yellow, we take wrinkled green. And we don't take round green, we take round yellow. But here the 2 new contrasting character or 2 new recombinant character are obtained here. 2 new recombinant character is obtained in F2 generation. 2 new recombinant characters. Okay. So here we can say that here 2 new recombinant character is obtained here. 2 new recombinant character is obtained here. Are you getting my point? Are you getting my point? That
Yes,
two
sir.
new recombinant characters are obtained here that we have not taken. And this is the only thing that we get in the dihabit cross. Okay. So please make it properly. Then we're going to discuss. Okay. The 2 new recombinant characters are obtained here. Done. Done this one. Note it fast. Done, Jaslin? Madiha?
Yes, sir.
Yes, sir.
Okay. Let's move further. So this is the 2 new recombinant characters are obtained when we are just going to how it is possible. How it is possible. Now everyone has to concentrate here that how it is possible. Okay. Let me explain you. Okay. First of all, you need to make it clear here that round shape is dominant over wrinkled shape. That's why we take capital R for round and small r for wrinkled. Okay. Round is dominant over wrinkled. That's why we take capital R for round and small r for wrinkled. Like in the previous one, we were taking, since tallness is dominant over dwarfness, so we take capital T for the tall and small t for the dwarfness. Similarly, in F1 generation, we find that when we cross round yellow with wrinkled green, we get all round yellow. So round and yellow is dominant. So for yellow, we take capital Y, and for green, we take small y. Got it? Got it? Got it? And if we want round, if we want round shape, then round shape can be obtained by capital R if both are capital R, or if we have capital R and small r. 1 R is sufficient. 1 R is sufficient to obtain round shape. 1 R is sufficient to obtain round shape. Are you getting a point? Tell me.
Yes, sir.
One R, because like in capital T, Tallness can be obtained by capital T, and capital T and small t, we can get it tall. So similar is, this will gives you round shape, this will gives you round. Similarly, wrinkled shape can be obtained when we have small r, because wrinkled is not dominant. So if we want, like we want to get the dwarf, so we need same small t, small t. Similar cases for if we want to get those character that is not dominant, we need both same small r, okay. Similarly, yellow can be obtained by capital Y or capital Y, small y. And same green color can be obtained, will be obtained only when there is both small y. If there is even one capital Y, it will give yellow color. So please draw this one, and make note this one. Note it down fast. Try to write it fast. Done? Noted? Noted this one? Should I move? Okay. Next is, now how we get this one. This is very important that we need to understand that how we are going to get this value, okay, and how we are going to get this one in the actual form, that how we are getting 9 round yellow, 3 only one wrinkled green, 3 round green, and 3 wrinkled yellow. So how we are able to get this one, let me explain you. In dihybrid cross, so you have to make this table like this way, okay, and then we will have 3 lines, 16 column for 3 lines will be there like this, and 3 lines here like this. Okay, now we have round, we will have like this. First we will write, the way of writing this one is very easy. First write capital R, capital Y, and here also capital R, capital Y. Then write here small r, small y, then write here small r, small y. Then write capital R, small y here, capital R, small y, and here small r, capital Y. Same here, capital R, small y, and small r, capital Y. Got it? Got it? Are you getting, Jobita, Jaslin, Madhya?
Yes, sir.
So here, how will you able to get? When you multiply these two, these are the gene. Like we have capital T, small t, then they are going to fuse with this one. You will get capital T, small t. So here we are going to have capital R and capital R and capital Y, capital Y. When you multiply these two, okay, you will get capital R, and capital Y, small y. Then again capital R, small r, capital Y, then capital R, small r, capital Y, small y. Okay, then capital R, then capital Y, small y. Okay, then capital R means this is multiplying with this one. Okay, capital R, small y, then again capital R. Okay, got it? Are you getting my point? Capital R, capital Y, small y. Means this is multiplying with this one. So one by one, each and everything is multiplying here. Okay, are you getting? A small
Yes,
y.
sir.
This one is capital R, small y, small y. Then capital R, capital Y. Then capital R, capital Y, small y. Then small r, capital Y, capital Y. Then small r, small y, capital Y, small y. Then capital R, capital Y, small y. Then capital R, small y, small y. Then capital R, capital Y, small y. Then small r, small y, small y. Are you getting this one? This is the way how we are just going to write. Got it? Got it? Now what we are going to do here is that you need to understand one thing, that for round yellow, round and yellow, round yellow color, round yellow. Okay, for round yellow, if you want to obtain round yellow, what you need? At least round shape, you need at least one capital R, because round is dominant over wrinkled. And for yellow, you need at least one capital Y. If both are capital Y, then it is round yellow. If it is small r, this is also gives you, means at least one capital R and one small capital Y is being required. And all the and this whole line is going to have capital R, one capital R, at least one capital R and one capital Y. All are going to have this one. This is also going to have one capital R and capital Y. This is also going to have one capital R and capital Y. This is also going to have one capital R, capital Y. This is also going to have one capital R, capital Y. This is also going to have capital R and capital Y. These all are 9 shapes. 9 are going to have, means if these genes are present, then they will give you round yellow. So how many round yellow you are going to get here? Tell me. The round yellow will be 9. Okay. Then for round green, round shape and green. So at least round should be there. One capital R should be there. And for green, since green is not dominant, so then we should have both small y. So at least one capital R and both small y will gives you round green. So how many can give you round green? Round green. So one capital R and both small y. So one capital R and both small y. This will gives you round green. Okay, this will gives you round green. This will also gives you round green. Both capital R, at least one capital R and both small y. And this also gives you round green. So these are the 3 round green that you will get, round green as 3. And then wrinkled yellow. Okay, then wrinkled yellow. So wrinkled yellow, for wrinkled shape, both should be small r. Both should be smaller, because wrinkled is not dominant over round. So if you want to get wrinkled, the both should be smaller, and one should be at least for yellow. If there is one capital Y, then it's okay. If both capital Y, it will be yellow. But if it is only one Y, then it is yellow. So both small r and one capital Y will gives you round yellow. Then both small r, one capital Y is here. This will gives you wrinkled yellow. So this is wrinkled yellow. This is wrinkled yellow, and this is also wrinkled. No, both small r and one capital Y. Actually, this should be, I have written here capital R. This should be small r. It should be small r, because this is small r. This will give you small r. So this will give you small r. Now you can check it. You can write it. This will be wrinkled yellow. So this will be 9. 3 will be the wrinkled yellow, and 1 will be the wrinkled green. Only for wrinkled green, because wrinkled is not dominant and green is also not dominant. So if you want wrinkled green, then they both should be for wrinkled green, because wrinkled is not dominant, you have to write small r both. And for green, you have to write capital Y, small y, small. Okay, got it? So this is wrinkled green. And for wrinkled green, we're going to have both small r For wrinkled shape, because wrinkled is not dominant over round, so both should be small r, and for green is not dominant over yellow, so both should be small y. So this is only one, wrinkled green. Got it? Got it? So this is the way that how we get this one in the F2 generations. And this is only possible how we are just going to get a new recombinant character here. How we are just able to get the new recombinant character, and how we are getting this one. So let you understood here. Are you getting this one, Jobita? Jesslyn? Are you getting? Madhya?
Yes, sir.
And you, Jobita? Are you there? Turn on your camera, please. Turn on your camera. Let me check whether you are studying or not. Okay. Now, next is you need to understood that we are getting this one. Okay. So this, have you drawn this one, Jobita, Jaslin, and Madhya? Have you drawn this one?
Yes, well done.
This is an important table. Okay. Now you need to know here that we have taken round yellow and wrinkled green, but we get 9 round yellow and only 1 wrinkled green, and we were taking the round green and wrinkled yellow. These we have not taken. These we have not taken. Okay. We have not taken these. Okay. Got it. These two we have not taken. Round green we have not taken, and wrinkled yellow we have not taken. We have taken only round yellow and wrinkled green. But the two new recombinant characters are observed here. 3 round green were observed and 3 wrinkled yellow were observed. This we have not taken. So how it is possible? This is only possible if we have a separate gene. This is only possible if we have a separate gene for round shape and the yellow shape, and wrinkled shape and the green shape. Just for example, let me explain you here that, for example, this is a gene that control round shape, and this is a gene that control yellow color, and this is a shape that control wrinkled, and this is shape, this is a gene that control green. Okay, just keep in mind here, this is very important. This is very important that what is the observation or what is the conclusion of Mendel dihybrid cross? Conclusion of Mendel dihybrid cross. What is the conclusion of Mendel dihybrid cross? So the conclusion of Mendel dihybrid cross is that there is a separate gene, because we are just obtaining the two new recombinant character that were not taken. This is only possible if we have a separate gene for each trait. For round, there is a separate gene. For round, along with we have yellow. For yellow, we have different gene. Okay. For green, we have different gene, and for wrinkled, we have different gene. Now they can rearrange themselves. Now it is possible that they can rearrange themselves. Okay. This is possible. Like this can goes to this one, and this can comes to this one. Okay. So the new two recombinant characters are only possible if if we have the two separate genes, and they can freely assort themselves. They are free to assort themselves, means they can assort themselves in any order they wish. They can go. It is not bounded that every time the round gene will remain with the yellow. No. Okay. No, it is not fixed. Round gene can go with the green color, and yellow gene can also go with wrinkled color. So it is only possible when we have a separate gene. For round shape, there is no specialized gene, or that is like this is, I'm just making here. Let's imagine that there is a gene that is controlling both round as well as yellow. There is no any kind of gene. No. We have separate gene for yellow, round, and for yellow we have separate gene. There is not a single gene that is controlling the two characters or two traits. There is not a sep. We have the different genes. Got it? Got it? Understood this one?
Yes, sir.
So please note it down. This is called law of independent assortment. And what does law of independent assortment states? The law of independent assortment states that for every trait, there is a separate gene. For every trait, there is a separate gene and we called it factor. Factor in terms of Mendel's language. Mendel. There is a separate factor, separate gene, and it is free to assort itself independently. Note it down. Note it down fast. And it is free to assort itself independently. For every trait, there is a separate gene and it is free to assort itself independently. Note it down. Okay. Done. So next question comes in examination that how do Mendel experiments show that traits are inherited independently? So traits are inherited means they can be inherited and independently they can assort themselves. So Mendel experiment in dihabet cause we have seen that there are two new contrasting characters were two new recombinant character were observed that we have not taken, and this is only possible if we have the separate gene. Okay, so please note it down that how do Mendel experiments show that traits are inherited independently. So note it down in Mendel experiment, Mendel's dihabet cross experiment, the two new recombinant characters were seen which were not taken, i.e. round green and wrinkled yellow, that we have not taken. This were wrinkled round green we have not taken. We have taken round yellow, and wrinkled yellow we have not taken. We have taken wrinkled green. But the two new recombinant characters are observed. This is only possible. It is only possible when there is a separate gene. When there is a separate gene for each trait and they are free to assort themselves independently. Note it down fast. Are you getting this one, Jobita? Jobita, are you there in the class?
Yes, sir.
Yes, sir. I'm just asking you so many times. Why not responding? Again, she got.
Sir, I am here.
Okay. Do this one, please. Noted this one? Jaslyn, noted?
No, sir.
Noted fast. Done? Jaslyn, done?
Yes, sir.
Madhya?
Yes, sir.
Okay. Next is blood group. Okay. Before the okay, let's have this blood group. So if I talk about the blood group, we have blood group A. A blood group is only possible if we have IA and IAIO. This is homozygous A. This is homozygous A, and this is heterozygous A. Heterozygous A. Okay, this is B, the same homozygous B, heterozygous B. AB is blood group is only obtained when we have A and B allele, and when we have O, this is IO. Then we're going to get the B blood group. Okay, got it? O blood group. So here, if the question comes in examination that a man marries, this is a question that is asked in the NCRT. Okay, in text question, a man marries a woman with blood group A. With a man with blood group A marries a woman with blood group O. Okay, and their daughter has a blood group O. Their daughter is going to have blood group O. Is this information enough to tell you which of the trait or the blood group A or O is dominant? Is this information enough? Okay. So if I ask you that a man marries a woman with blood group a man with blood group A marries a woman with blood group O, and their daughter is going to have, man is going to have blood group A, and woman is going to have blood group O, and their daughter is going to have blood group O. Is this information enough to tell you which is dominant or not? No, actually A is dominant over O, but here this is not sufficient data. This is not sufficient. Okay. How we can say this one? That for condition A, we have IA, and we have IAIO. This is for A. And for condition O, we have IOIO only. So when the condition 1 is there, that man is going to have homozygous A, and women is going to have O. O is always homozygous. So here we will have IAIO, 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 and IAIO. So in all these case, we will have almost everyone, means IAIO will gives you blood group A. This will also give you blood group A. This will also give you blood group A. This will also give you blood group A. So in condition 1, we will say that when father is going to have homozygous A, then there is 0%. Then there is 0% chance 0% chance that the daughter will have blood group O. Daughter will not be going to have blood group O because if the father is going to have homozygous A, then in that case the daughter will not have any. I mean, there is no chance of getting this one. Are you getting? Tell me.
Yes, sir.
Okay. Next is, and if the father is going to have heterozygous O, then in that condition, you will notice here that this IA and IO and this IA and IO. Okay, this when this will meet together, will get IAIO. This will get IOIO. Then O and A, IAIO, and then this two will gives you blood group O, and this two will gives you blood group O, and here it will gives you blood group A. So there is 50-50% chance. So in condition 2, we have 50-50% chance that the father is going to have, daughter is going to have blood group O. So in this case, we cannot say that data is sufficient that we can tell that which is dominant or not. Okay, got it? Got it? Got
Yes,
it?
sir.
Okay. So please take the screenshot of this one. This you have to note it in the homework. This question you have to do in the homework. Okay. So please note this one. We'll send you this question in the homework. Take the screenshot of this one. Taken? Take this one also. Jaslyn, done?
Yes, sir.
This is an important question from NCERT blood groups. Taken this one? And this one also. This one, these questions are also, this questions answer is also provided in your notes that we have sent you, handwritten notes. Okay, that you can check in the group. We have provided you the handwritten notes. Now, the next question is the sex determinations. Okay, in some animals, if I talk about the lizards and the like in lizards, like in human females. Okay, sorry, not in human, in hands. Talking about uh, actually there is temperature. Temperature can also results in change in the gender. Okay, the incubation temperature. For example, these are the hens. These are the eggs that are laid by the hen. Okay, and she is sitting over it. So the temperature, high incubation temperature can result in the female, and low incubation temperature can result in the male. But this is opposite in the lizards. Okay, so we can talk it that there is some, for the sex determination there can be environmental factor also, means temperature, and in human males or in animals like human beings, there is genes also, genes that are responsible for sex determination for the male and female child. Okay, so please note it down. Sex determination, that in some animals, the incubation temperature at which the fertilized egg are kept decided the gender. Okay, so in machines, like in pottery farms, we have the eggs, we just kept it at the particular temperature, then we're going to have all chickens. All chickens are there. And similarly, if we put them at higher temperature, then we will going to have all hens. Okay, the incubation temperature results in the gender, okay, of male and females. So note it down that in some males, in some animals, the incubation temperature results at which the egg is fertilized decide the gender. Like example, hens, lizards, lizards, and genetic. It can be genetic, that in some animals like human being, gender or individual is determined by a pair of chromosomes called sex chromosomes. Note it down fast. Done. Tell me, done. Jaslyn, Madiha,
Done, sir.
done.
No, done.
I
Yes,
will
sir.
check. I will check the notes. You need to send me the notes whether you are writing or not. You have to need to send me the copies of the notes that you are writing or not. Okay. After the class, you have to send the notes. I will check it. Okay. Now, how it is possible? Let me explain you this one. That there are 44 autosomes. Like in human beings, we have 46 chromosomes. Out of this 46, we have 44 are autosomes. And in human males, if I talk about human male, and this is female, human female, we have, okay, human male and human female. human female. We have 44 autosomes and X and Y chromosomes. X and Y chromosomes. These are known as sex chromosomes. The 44 are autosomes and X and Y are sex chromosomes. Here also in human female, we have 44 autosomes and XX as sex chromosomes. Okay, in human females. Okay, so when you know that all the cells are diploids except the gametes. So when we have 46 chromosome, then the gametes are going to have 23. Okay, this 23, out of this 23, we have 22 autosomes, and one is in human males. In human males, we are going to have one X. Another we can have 22 autosome and one Y. So we are going to have 46 as 44 as autosomes and one X and one Y. So when they are going to divide, separates into 23, then we're going to have 22 as autosomes and one X, and another will going to have 22 as autosomes and one Y. Okay. And human females have also 46 chromosomes, out of which they have 44 as autosomes and XX as sex chromosomes. And this then man going to have the eggs. So since they are going to have the same kinds of egg, they are not going to have different kinds of egg because they are going to have XX only. So they're going to have 22 as autosomes, 23 as egg in the egg, and in the egg we're going to have 23 in the egg or ovum. Okay, and this is going to have 22 as autosome and one X. So when this and this is going to fuse, then it will gives you 44 as autosome and one X, another X. And when this is going to fuse with this sperm is going to fuse. This is the chromosome of a sperm. Okay, so if this sperm fuse with the egg cell, then it will gives you 44 as autosomes and one X and one Y as a chromosome. This will result in a female child, and this will result in a male child. Okay, male child. So the question comes that how these six chromosomes is helping you, or in the helps you to determine the sex of a child. And males are only responsible because males have different types of sperm. So only the sperms, if the sperm of X chromosome fuses with the ovum, then it will be a female child. If the sperms of Y chromosome fuses with the ovum, then it is a male child. So this is, you need to keep in mind that males are only responsible for the uh, sex of a child, not the females. So in our societies, generally the females are blamed that if they are not having the male kids. Okay, so they are generally blamed that they are going to have only female. So this the males are only responsible for the uh, sex of a child, not the female. They should not be blamed for the sex of a child. Okay, noted. Have you drawn this one? Draw this one, please. Draw this one.
Done, sir.
So this is important. Have you noted this one, Jaslin, Madiha?
Yes, sir.
Yes, sir.
Okay. Now the question comes that if the question is being asked in the examination that how it is responsible, or how the males are responsible, or how can you determine this one? So note this one, note the question, note this one. Chromosomes. Note this one. In human beings, there are 23. In human beings, there are 23 pairs of of chromosome dome. Out of these 22 chromosomes are autosomes, are called autosomes, are autosomes, and the last pair, and the 22 pairs. 22 pair means 44. 22 pairs. Chromosomes are autosomes, and the last pair, is called sex chromosome that help in deciding the gender, the gender of that individual. Okay. In the above diagram shows that half the children will be boys, and the half will be girls. All childrens are will inherit X chromosome from their mother, regardless whether they are boys or girls. Okay. Thus, children and if she is a girl, in our word, show that the children wise will all children will inherit X chromosome from their mother, regardless whether they are the girls or boys or girls. Okay. and boys will get Y chromosome will get Y will get Y chromosome with from their father from their father, and girls will get X chromosome their father. Thus, thus that father are responsible. Father are responsible responsible for sex of a child, not mother. So many times this question is being asked in the examination, so please note it down properly. Okay, and the questions that will be sent in your homework that you need to do. That there's so many types of questions being asked in the examination on this topic, that how father is sexually determined, how fathers are responsible, how women are not responsible. So in case study questions, there are so many questions being asked on these topics. Okay, so everybody understood this concept, and have you noted this one? Tell me, have you noted?
Yes, sir.